I want to welcome everybody to the College of Engineering Charles DeLisi's Distinguished Lecture. That's an annual lecture endowed with a generous gift by Charles DeLisi. Charles, as many of you know, was the dean of the College of Engineering, 2 deans before I was, who completely transformed the college into a major research college of engineering and really propelled us as a college to embrace the power of research to become a world-class university and a world-class college of engineering. He, of course, on his own is a world-class scientist and has had a huge impact on science. He's really commonly called the father of the Human Genome Project, and he is the person and the only person to receive the President's Citizens Medal for having started that as a government-wide genome project. And the impact on medicine is just extraordinary and happening in greater leaps every day. So we're deeply indebted to having a lecture on the scientific accomplishments of one of our own faculty and alumni to be named after Charles DeLisi. So Charles, stand up, please, and let's just honor your presence and your gift to make this possible. Okay, so we have first an award for the early career excellence. This award goes to a junior faculty, assistant professor faculty, who has built an extraordinary record early in their career of excellence and impact in their field with a clear sense that they're going to propel their research to an area, to a space that truly transforms their discipline. We give this every year, and this year the award will be to Sahar Sharifzada. Let me tell you a little bit about her. Sahar received her bachelor's in electrical engineering and computer science from Berkeley, her master's and PhD in electrical engineering from Princeton, and did her postdoc fellowship at Lawrence Berkeley National Laboratory. In 2014, she joined Boston University as an assistant professor of electrical and computer engineering and of material science and engineering. I think she is one of the first, one of the early people we recruited at the nexus of the division and the department. Her research area is to advance our understanding of the optoelectronic properties of materials using computational methods to advance how electronic and optical properties of non-standard semiconductors work. Her work is focused on excited state phenomena, semiconductor physics, electronic structure theory, and computational material science. She's published 28 peer-reviewed articles to date that have been cited 1,250 times and has an H-index of 17. Already 4 of Sahar's papers have been cited over 100 times. She's given 30 invited talks at other institutions and conferences, and she's been funded by the U.S. Department of Energy via an Early Career Award and a 3-year Division of Material Research grant from the National Science Foundation. Most recently, Sahar was selected as a Rising Star of Science by Nature Index. This is a 2018 award. Here is a portion of the quote from Nature as to how they decided who gets this award in general. From cutting the cost of solar electricity to reducing the risk of ovarian cancer, the 11 early to mid-career scientists profiled here, this is in the magazine, are emerging as leaders in their fields. They stood out from among 500 scientists assessed using the power of the Nature Index and the League of Scholars whole of web rankings. They are bringing fresh ideas in a range of disciplines from cognitive neuroscience to geology and condensed matter physics. Their initiative, curiosity, and flexibility have given them an edge in a competitive research environment. So please join me in congratulating Sahar for winning this early career award. I think I just used up all the time for the DeLisi lecture. Okay, so now we introduce our distinguished DeLisi lecturer for 2019. Chris Chen received his bachelor's in biochemistry from Harvard, his MS in mechanical engineering from MIT, and his PhD in medical engineering and medical physics from the Harvard-MIT Health Science and Technology program. He earned his MD from Harvard Medical School. Chris was previously an assistant professor in biomedical engineering and in oncology at Johns Hopkins University. The Skirkinich, I can't say the guy's name. He had a named professorship of innovation and was the founding director of the Center for Engineering Cells and Regeneration at the University of Pennsylvania. In 2013, Chris joined BU as a distinguished professor of biomedical engineering. It took me only 10 minutes to recruit him. It's a 2-year process. It was all worth it. He's the founding director of Boston University's Biological Design Center, director of the Tissue Microfabrication Laboratory, and professor of biomedical engineering at the Wyss Institute for Biologically Inspired Engineering at Harvard University. He's also the deputy director of the NSF ERC on cellular metamaterials. He has been an instrumental figure in the development of engineering cellular microenvironments to understand how cells build tissues. His group pioneered the use of micro and nanofabrication technologies to identify the underlying mechanisms by which cells interact with materials and each other to build organized tissue, and to apply that knowledge to biology of stem cells and tissue vascularization. He's arguably the world leader in determining how the mechanical and chemical environments of cells determine their fates. He has over 220 peer-reviewed publications, and his papers have been cited over 44,000 times, and he has an H-index of 96. He has 7 papers cited over 1,000 times, of which he's the lead or senior author on 5 of them, of those 5, 1 has been cited over 4,000 and 1 over 5,000 times. He's given 91 invited lectures, 43 of which have been plenary or keynote lectures at a conference. He's served as an editor for the Journal of Cell Science and the Journal of Biomedical Devices, and has been on many editorial boards, including Science Translational Medicine and the Journal of Cell Science, as just some examples. He's also won many awards. He's a fellow of the American Institute for Medical and Biological Engineering. He was on the Board of Trustees for the Society for Biomems and Biomedical Nanotechnology, and a Defense Science Studies Group member. He was awarded the President's Early Career Award for Scientists and Engineers, often called the PCASE Award, the Angiogenesis Foundation Fellowship, the Office of Naval Research Young Investigator Award, the Mary Hullum George Award for Biomedical Research, and the Herbert W. Dickerman Award for Outstanding Contribution to Science. He's chaired keynote and symposium at Gordon conferences. And most recently, Chris was informed that in 2019, he will receive the Biomedical Engineering Society Robert Pritzker Distinguished Lecture Award, which is the BMES's most prestigious honor. His talk today is entitled, How Complex is Simple Enough? Engineering 3D Cultured Models of Physiology and Disease. Please join me in welcoming and congratulating Chris Chen.
I'm going to have to cut half my talk out. Ate up all my time, Ken. Well, first, I also would like to thank the School of Engineering and BU and Charles DeLisi for supporting this event and providing the inspiration for this whole concept. I'm deeply honored to be joining the ranks of previous distinguished awardees, and I'm excited to share with you, with everybody in this community, what we're working on. I'd like to start with a brief history of how we got interested in this particular question and share a few ongoing stories of what we're working on. Let me start by just talking a little bit about cells in the context of ways in which we interact with mammalian cells. One is cells that are in culture dishes, and one is cells that are sitting in the body. And what I'd like to tell you, no big surprise, when we take cells out of the body and we study them in the dish, they hardly represent in almost any way whatsoever what's going on with cells that are in the body. So the question is, why? Why are they functioning differently when you take them out? And the main reasoning has been that the milieu that the cells are sitting in is different when they're sitting on a plastic dish than when they're sitting on other cells that are in your body. And when we think about the milieu, the main focus has been on a series of proteins and lipids that the cells are exposed to that we call growth factors or cytokines. And I'd like to argue that in addition to those, the insoluble environment, how they interact with the physical structures that are around them, have a major effect on what they do. So when we think about those physical interactions, we think about interacting with the solid extracellular matrix that the cells are sitting on, the neighboring cells that they make direct contact and adhesion with, and the mechanical forces that they experience as they go through life. So one of the questions that I started out my own lab with is how important are these structural and mechanical cues in affecting cell function? And I'll go through just a couple of really brief slides. This is something that we were really interested in very early on, which is simply that if you look at cells that are cultured in a dish, they proliferate very rapidly until they become confluent. Once they sort of make contact with each other, they sort of get squeezed in, and then proliferation shuts down almost completely. Now, in that setting, the media or the growth factors that they're sitting in are the same at the beginning of the experiment or at the end of the experiment. You can wash it and you can put in fresh growth factors and they still don't grow anymore. So the question that we were interested in is, what is it they're paying attention to? What is it that they are receiving as a cue that's changing their behavior? And the two things that we thought were, well, obviously when the cells get crowded, they're making more contact with their neighbors, and perhaps that does something to shut down proliferative signaling. And the other is that they're making less contact with the underlying substrate, in this case plastic, because they're getting squeezed together. So they sort of see less plastic as they go. My advisor, Don Ingber, and his advisor, Judah Folkman, had this idea that it had something to do with the contact that the cells made with the underlying substrate. And Judah had published a paper in Nature suggesting that it had to do with how well the cells could flatten out against the surface. So I'm going to fast forward over about a decade or two of research, my PhD work, some of the work that we did when I first started my lab, and I want to summarize in this one slide. And that basically is that you can drive changes in cell proliferation signaling by a number of different ways that seem to sort of drive the same functions. So what I'm showing here is a bar graph. I'm going to see if I can get this to show up as a pointer. You can see this bar graph here, where what I'm showing is luciferase activity off of a promoter, which is the serum response element. So when you stimulate cells with serum, one of the first transcription factors that gets activated then binds to the serum response element and drives transcription. And the proteins that are expressed during this are the ones that are important for cell cycle progression. So what you see is that when you take cells that are not confluent versus cells that are confluent and you stimulate with serum, what happens is that the amount of activity of that promoter decreases. It also turns out that if you take cells and you put them down all at low density, but onto dishes that are coated with different amounts of matrix ligand, then you get the same effect. So if you have less ligand, the cells adhere less to the surface, and you hit them with serum and they don't seem to respond, and you can see that over here on the right bar graph, this bar versus this bar. I had done some of my work in micropatterning, and what we had done was we had coated substrates with a high density of fibronectin ligand that we knew would be enough to cluster receptors. And when we do that, if we pattern it so that the cells are sitting on these small spots so that the cells will attach, the receptors will cluster, but the cells can't flatten out against the surface. Again, if we hit them with growth factor, they can't drive this transcriptional signaling response. And then the last thing I'll show you is that if we put them onto substrates that are of different stiffness, substrates that are very soft, what happens is that when the cells are sitting on those soft substrates, you hit them with a growth factor, again, they don't turn on these key transcriptional signals that drive proliferation. And in all of those cases, the cells are arrested and they stop growing. So this suggests that there's some kind of interaction inside the cell between cell-cell adhesion, cell matrix adhesion, mechanical forces, et cetera, that drives this response. And I'm going to show you just a different model system of how we sort of figured this out. What we did was we were looking at a cell known as the mesenchymal stem cell that we can isolate from human bone marrow or human fat. And these cells have the potential to differentiate into different lineages, mostly of the musculoskeletal system. What we found is that if we plate these cells onto substrates where the adhesion signal is very low, they'll undergo adipogenesis. They turn into fat. And if they attach on surfaces and get a very strong adhesion signal, then they turn into bone. And shown here on the top right is just a picture of cells sitting on patterns where we have, again, small islands where they can adhere but can't spread out, versus if they can spread out very well. And they will switch between bone and fat differentiation under the same conditions. We established a tool to be able to measure the forces that the cells generate against substrates, because we had thought, based on those earlier studies I showed you, that cells were generating forces differently when they were on highly adherent or less adherent surfaces, and that they were using those forces to transduce those signals. And so the way we made these is we made these sort of silicone rubber pillars, and we could coat the tips of the pillars to be adhesive for cells and the rest of it not. So when you plate cells, the cells would spread across those pillars, and then as the cells crawl around and pull on them, the pillars will bend. And so just based on a simple microscope image, you can back out the forces that the cells are generating at every adhesion point that they make with the substrate. So using those tools, what we found out was that when the cells could adhere and spread against the surface, they would increase their force generation against that surface. And it turned out that that force generation was important for their differentiation. So in the sort of biological stick figure world, this is the way that we think about the control system. So as the cells can spread out, it turns out they activate this signaling cascade that I'm not going to go into the detail of. This Rho GTPase activates, and when it does, it turns on myosin. And myosin then causes the cells to contract against the substrate. When it contracts against the substrate, then we increase stress at the cell surface interface. That stress turns out to activate the adhesion signaling pathways that then drive bone differentiation. And if you block that contractility, then they undergo fat differentiation. So this is another way of sort of describing that control system. Again, I'm not going to go into the details of it because I just want to get to a couple of main points for this. We then found that if we took these cells and exposed them to different soluble factors, we could get different differentiation responses. So what I showed you earlier was a response that was driven by dexamethasone, which is a glucocorticoid stimulator, that would drive them between bone and fat, depending on, well, how adherent they were. It turns out that if you use BMP, another growth factor, they would switch between bone and cartilage. And with TGF-beta, they would switch between muscle and cartilage. And so what we were seeing, I think, is sort of like a combination switch. They were paying attention to different growth factors and different adhesion signals and sort of using that combination to decide what to differentiate into. So I think of this as sort of like a simple combination switch. It turns out that with different cell types, you get different responses. So these three in the middle are all with these stem cells. If we take epithelial cells and we hit them with TGF-beta, they'll switch between what we call EMT, or epithelial to mesenchymal transition. They become fibroblast-like. And when they're not adherent, they undergo apoptosis. So the point here is just saying that not only do different combinations of inputs give you different outputs, but depending on what cell type you have, you can drive those same inputs to give you different kinds of outputs. Okay? Okay, now quickly transitioning. We asked ourselves, all the work that we had done was on 2D at this point. We got a lot of criticisms from grant reviewers, and I always ignored them, that bone doesn't occur and fat doesn't occur on surfaces. It happens in three-dimensional tissues. So we said, okay, So let's start developing some tools to be able to ask what happens when we embed them within matrices. We developed some matrices where we could vary the stiffness of the gel as a way to kind of vary this adhesion signal. And what I'll show you is just one piece of data. This is a complicated slide, so all you need to do is just look at those two color pictures. We had a gel that was soft, that when we embedded cells into it, the cells would kind of spread and dig through the gel, and they kind of spread out into it. And as we stiffened the gel, what happened is that the gel became harder and harder for the cells to spread into. Right? You can think of it as to spread into it, it has to cut through those cross-links, and it was getting harder and harder for it to do that as we stiffened it. So then the question we asked is, these were mesenchymal stem cells, do they differentiate into bone or fat in these conditions? And what we found is that the ones that were in the stiffer gel turned into fat, and the ones that were in the softer gels turned into bone. So this is the way I sort of describe this. On 2D, we showed you that if we make the surface stiffer and stiffer, cells adhere better, they generate more stress against that surface, and they increase their bone production. In 3D, as we increase that stiffness, they become more restricted. They're not able to spread into the surface. They don't interact as well against the substrate, and they undergo adipogenesis. When we found this result, it took me about a year to publish it because all the reviewers cited all of our previous work, saying that we must be wrong. So what we had to say was we were wrong, but it's not the new study that was wrong. It may have been the old studies. So this led us to something that made me change the entire makeup of my lab. We had been studying for 15 years how forces affect cells, and we came up with what we thought made sense. And then what we realized is that depending on how you configure your culture, you get completely different responses. And it doesn't, you might say, well, why would you worry about that? And I think the way we thought about it is, well, if we don't know how to put the cells in a context that is very similar to the way that they exist in your body, then it's really hard to interpret whether what we're seeing is actually the way that those cells would interpret those signals when they are in your body. Okay? So this isn't just about dimensionality, 2D versus 3D. It is also true when it comes to other aspects of what I think of as architecture, how the cells are organized. So you see here some pictures of cells that are in 3D, like this, one cell that's embedded by matrix, or a group of cells that are surrounded by matrix and they're polarized and they have sort of a lumen in the middle, right? That structure has different signaling properties. And has different signaling properties because the cells are interacting with each other. There's an apical domain sitting inside this lumen that has different growth factors that they're exposed to. Different receptors are trafficked to this side versus this side. So the way that a cell interprets, say, force here is going to be quite different than the way it might interpret it here. So that leads us to this worry that we better get the cell structures right if we're going to go ahead and try to study them out of the body. So this was our aha moment. Now, of course, the reality is that your grants fund you to do the things that you were working on, not the things that you're going to be working on. So it took about 5 years to make that transition as you start getting data that lets you then get grants in the new area. So what we want to do is build tissue-like structures. That's the goal. That's our current goal. And so the first question is, how do cells do it? How do they make tissues, and can we use that to sort of mimic those processes in our own lab? And related to that is that there's sort of bottom-up approaches, things where the cells are going to drive that patterning process, and then there's top-down tools that they don't have that we have that we can sort of force them into certain initial conditions and ask what that does. We know that forces turn out to be really important in driving most of the morphogenetic changes that occur in the body. And we'd like to be able to control those or take advantage of those for various applications. And keep in mind that our final goal is to get the organizations right so that these cues, the cell matrix, cell, the mechanical forces, are in some way similar to the kinds of experiences that those cells would have when they're in the body. And if we get those right, then voila, we have an in vitro culture that we really believe models what happens when we're trying to study it in the context of human physiology. So I'm going to tell you a couple of stories of what we've been working on, mostly in two contexts. One is in muscle, heart muscle, and the other is in trying to build a slightly more complicated 3D structure that I'll say is building compartments, vasculature, lumens, et cetera, and sort of seeing how those can contribute. So in the context of building muscle, this is the first system that we developed, and we've been sort of building on this. We know that when we take cells and we embed them into a matrix, the cells will spread into the matrix and try to contract it like a ball. And what we found is that if we simply took that matrix plus cells and put it into a well where we have pillars in those wells, then the pillars sort of catch the matrix. And you can see that in this movie that I think has been cycling. So what happens is that as soon as those cells are sitting in the gel, they start to contract and compact that gel. Because those pillars are there, they get caught, and then the whole tissue sort of forms this rubber band around these pillars that have captured it. Because they contract, what happens is that if it's a fibrous matrix like collagen or the like, then those fibers get aligned in that direction of stress. And when they get aligned, the cells align. So we have this sort of aligned tissue structure. Here, what we did was we took the same technique, what I showed you before was fibroblasts, and we put in cardiomyocytes. And what you see up here at the very top is cells that are just seated in here, and you can see that there's some sparks and beating that is totally disorganized. What happens is overnight and after a few days, that thing compacts, the cells get more dense, the cells spontaneously make mechanical and electrical couplings with each other and form what we call a muscle syncytium. The whole thing becomes connected. And when that happens, this is what happens. You see this spontaneous beating of this whole muscle as if it's a whole unit. And because we make these pillars out of an elastic rubber, then we can sort of look at the force that it takes to contract, and we can measure what you think of as sort of a relaxed state and a contractile state in these tissues. So what do we want to do with these? We want to model heart tissue, and what we started doing was working closely with collaborators, Cricket and John Seidman, who are over at Harvard, and they really pioneered this area of sort of genetics underlying various heart diseases. So Cricket is a pediatric oncologist or pediatric cardiologist, and she's been collecting genomic data underlying diseases that cause dilated cardiomyopathies or hypertrophic cardiomyopathies in a number of children. Turns out that the most common mutation is in this protein called titin. Titin is a very large protein that is involved in forming what we call the sarcomeres, or the muscle units inside heart tissue. And so what we did was we took patient blood cells from these various patients, generated iPS cells, differentiated them into cardiomyocytes, and then put them into our tissue model. And what we found was that in every patient that had a defect in titin, we saw a major defect in their ability to form and contract tissue. And so you can see here, we have patients that have normal titin and patients with abnormal titin. And you can see major difference, obviously, just by eye. Of course, this could be a multigenic disease. There might be other mutations that aren't just titin. And so we also took the wild type patients and used CRISPR to knock out cause mutations in various parts of titin. And when we do that, you can see in this graph that basically all of our cellular CRISPR-mediated knockouts do the same thing. So one of the really cool things that we've been interested in is mechanical loading and its effect on heart tissue. And we've shown that if we take these heart tissues and we change the stiffness of the pillars, so we make the pillars stiffer, then the heart tissue has to beat harder to contract against it. And what we find is over time that they get stronger. This is what happens normally when we exercise. And it turns out that with these Titan mutants, they're not able to adapt to that increasing load. So we think that these patients have an abnormality that makes it hard for them to adapt in response to different kinds of stressors. One of the things that we observed was that the mutants produced a lot fewer sarcomeres, a lot fewer of these muscle units. So one of the things that we did as a follow-up to that story is that we started looking at the sarcomeres themselves. And one of the things that we observed that was really interesting is that if you take iPS cells, differentiate into cardiomyocytes, and you express a marker of the sarcomere itself, you can see that up here. These lines here show the sarcomeric pattern of these cells. We found that if you suspend the cells, all the sarcomeres disappear immediately. And when we replate them, the sarcomeres start reassembling. And so we're just sort of interested in how that process works. And you can see here, this is just a cell that's just been plated. And you can see that the cells are starting to form these muscle structures right before our very eyes. And they don't form randomly. They actually seem to assemble from these sort of points at the periphery of the cell and sort of stream inwards as they start to assemble. It turns out that this process of forming these sarcomeres relies on contractile power of the myosin itself. So we found that if you poison myosin, then the sarcomeres start to fall apart and are unable to assemble. And you can see this here. I'll just show you a movie rather than this graph. Here are two cells that we've patterned so that they're organized slightly differently. And you can see right in the middle of this, we add this myosin inhibitor called blebistatin. And you'll see almost immediately that those sarcomeres start to fall apart. And so what this shows you is how important that force itself is in assembling the very structures that are needed to generate power in these hearts. And it kind of makes sense why, if you start to have abnormal force generation, that you get out of you get into this vicious cycle of damage that is hard to break from. We then went on to show that the way that they assemble those sarcomeres is they assemble them, they seed them from the adhesion points between cells and matrix. And so if we stain for the points where the cells are adhering to matrix, that's marked by this protein called paxillin. You can see the cell is stuck on the surface at these points. And it's these very same points that you see this alpha-actinin, which is what starts the assembly of the sarcomeres, is coming from. And again, I'll show you a movie just to kind of give you a sense of this. So you can see that these sarcomeres are really assembling dynamically from the points of adhesion against matrix. So this tells us that the way that the cells organize their muscle-generating power depends on how they interact with the outside world, how they interact with the matrix, and that kind of feeds into this kind of molecular structure. So point is that we believe that force is, in this case, a key organizing link between structure and function from the molecular all the way to the multicellular scale. And I think I've given you a few examples of this. And it's based on this idea that then we assembled this team that is now the NSF ERC called CellMet. So for those of you that aren't familiar, this is a project that David Bishop is the PI of, and I'm involved in, that is trying to build this that heart at multiple length scales. And it's based on this idea that you need structure controlled all the way from the nanoscale all the way to the multicellular for that to work. Okay, I'm going to quickly transition to one other quick story just to give you a sense of another direction that we're going in. In addition to building muscle, we're interested in building vasculature. Vasculature is another thing that is really important for building different kinds of tissue, and we think that it's really important in this process. So what I want to do is just share with you a couple of pieces that we're working on. There's a couple of ways that we can imagine building vasculature in vitro. To us, vasculature's main function is to carry blood, right, and feed tissues. And so for that to happen, we need to have cells that are lining kind of these channels to then carry blood through those. We can build kind of top-down vessels, right? And shown here are just examples of a top-down vessel where we can take like a gel, stick a needle through the gel, pull the needle out, and then seed cells in it, and you've got a channel that is lined with endothelial cells, the cells that line blood vessels, and you've got something. But what we really want to do is build something that the cells can then reorganize over time, because we know that in our own bodies, blood vessels reorganize depending on the demands that they experience. So there's two ways that cells do it naturally. One is they undergo what we call angiogenesis, where they'll take an existing blood vessel and the cells will be sitting there, and then cells, let's say tumor cells or something else, is calling for new vascularization. They release factors, those factors go down to the blood vessel, the endothelial cells see them, and endothelial cells start to invade and build a new vascular bed in this new area. So that's called angiogenesis. And what we found is that we could take a device where we seed cells inside a pipe like I just described for you, and in a neighboring pipe we can put in angiogenic factors, and it triggers the endothelial cells to start to invade toward the other site. What's really cool is that depending on the combination of growth factors you use, you get totally different types of invasion. So you can get an invasion where the cells invade as single cells. They're not coordinated at all, and you get no vessels. But in certain combinations of cocktails, you can get the cells to invade as a continuous unit. And you can see here just a picture of that, where the cells at the tip are digging new channels through the matrix, and all the cells behind are basically forming like a luminal structure. And when they do that, they don't allow fluid to leak out during this time, so that they sort of protect the existing vessels. And then the other thing that you can do, that cells do, is they undergo a process called vasculogenesis, where instead of building new vessels from existing vessels, cells will simply invade into a whole territory, endothelial cells, and then at some point they decide to form a vasculature. I'm saying that because we really don't know what does it. They will all of a sudden find each other and connect into a giant web, and then they open up lumens inside them to connect them. And that's a process called vasculogenesis. So we have also modeled that process, and I want to just show you one example of that. So one of my graduate students, Greco Song, he basically found that if he took endothelial cells and mixed them with a stromal population, like a fibroblast, that they would spontaneously form networks that could carry flow. What was really remarkable is that if you take endothelial cells and you mix them inside of a channel with matrix, and they will find each other and connect and form what looks like a network. And over here, we have these two pipes that I showed you earlier where we seeded cells. They will connect to those pipes, but now if we flow a dye through those pipes, none of that dye goes into the middle of that channel. In other words, the cells are connected, but they don't open any channels for the fluid to flow through. But if he does this with fibroblasts and endothelial cells together, then you see what happens is they form the same network, but you see how it looks a little bit different? What happens is that these fibroblasts help to get the endothelial cells to sort of literally open channels in between them, so that you have sort of open cylinders. And now, when you flow fluid through it, a dye, that dye shows you that it's all connected. So we get basically a capillary vasculature. You can see these fibroblasts here in green kind of crawling around. It's very dynamic. It's an amazingly dynamic process. And after a while, this whole thing dies. The whole thing dies because the fibroblasts keep growing, and they eventually overtake this thing, and it becomes like a scar tissue. So for us, that's not very useful. We want to build a vasculature and keep it stable so that we can feed the other cells and things with it. So Greco found this really cool idea where he said, you know, if we take some of these tools that were developed in the synthetic biology world, for example, this one where you could trigger caspase activation, which kills cells, upon adding a small molecule. In this case, it's a small molecule that causes, it's a rapamycin-like analog, that causes these two domains to then cluster together. And what he did was he used that along with a caspase that is split. So the caspase is not active until this whole thing clusters. Now when you add this rapamycin analog, then caspase gets activated. So what did he do? He took that construct, put it into the fibroblasts. So the fibroblasts are perfectly happy. And he lets the fibroblasts help the endothelial cells make the network. And then at the end, he adds this rapamycin analog, and all the fibroblasts die. So I wanted to show you this movie. So on the left are the endothelial cells, the right are fibroblasts. It's the same cells. It's the same construct. And he adds this rapalog, and literally in a matter of hours, all those fibroblasts just go away. What he's found is that if he continues to monitor the vascular network is stable, and it's happy. And so we think we have a neat way to build vasculature. So the last thing I just want to share with you is that we're thinking about not just in vitro models, but how do we take our insights into clinical settings. So one of the things that we're really interested in is whether we can engineer vascularization in vivo. And we came up with this very simple idea that all of the work that's been done in vascularization so far has failed in the context of treating ischemia. And we think the reason why it fails is because when you inject VEGF or inject factors that induce VEGF or whatever, you get really great vascularization for a short while. And by great vascularization, what I mean is you can see lots of endothelial cells, you see lots of blood go into the area that you've injected. But after a couple of weeks, the whole thing fails. It dies, you don't get good blood flow, and the ischemic tissue never really recovers. And we think that what's happening is that when you get this massive invasion, you get vessels, but those vessels are totally disorganized. They're highly entangled. And so it's like Boston traffic circles. And that's not so good for real blood vessels, so we don't want those. So we said, well, what if we could use our patterning techniques to guide vasculature to form structures that are more organized? And to do that, we used some of these 3D printing techniques to make basically filament networks that we could dissolve away. And those filament networks could then act as sort of guides for where vessels could grow into a tissue. So I just want to show you the results rather than going into the details. So just imagine we 3D print some structure. We then fill that whole thing with an implantable gel like fibrin. We dissolve away those channels, so now you have open channels inside that fibrin. We seed into those endothelial cells, which we know will attract more endothelial cells, and then we implant it. And you can see here is a printed structure where we have these parallel channels with endothelial cells in it. This is a randomly organized structure with no organization whatsoever. Here's one where we've made a junction. This is in vitro. Then we implant it, and after about 7 days, then we inject a dye into the tail vein of the mouse and then image at the point of implantation. And you can see that we've got these new vessels now that are forming that are patterned based on what we've implanted. So you can see that the normal implantation, which is what people do now, you get completely disorganized, tortuous vessels that can't carry flow. But when we have this organized structure, we get really nice flow in these things, and they seem quite healthy. So is it any good? it turns out that you can take these patches, you can put them into different ischemic zones, like we've done this with rat hearts. We give them a heart attack, and then we can put these patches on with these organized vessel structures, and the organized vessel structures appear to enhance formation of new collaterals, and we can get recovery of hearts. We have also found that if we take that same idea and we co-seed those patches with liver cells, in this case human liver cells, this is with Syncytium, we find that, again, the vascularization is so good that it can support this implanted structure. So with that, I just want to say that along with what I showed you with the ERC, this area of trying to build sort of vascularized organ-like structures is something that now the VIS is very interested in helping to fund as a sort of translational objective. So we're hoping to sort of use what we've developed here at BU as kind of a starting point to go in that direction. So to conclude, I just want to say that we really believe that right now is a very important transition in how biology is studied. We have spent too many years studying cells on dishes and thinking that that's going to tell us what's going on in various forms of disease. And we've spent too many years studying diseases in mice. And for the same reason, we've been very ineffective at transitioning what I call basic science discoveries into translational meaningful results. And I think the reason why is because those models don't model humans. And we need to have better and better models developed that do model humans. I believe that if we actually can model the physiology appropriately, then it's pretty easy for us to think about how to use that to then, and we have lots of smart scientists around, to then figure out how to cure various things. So with that, I want to tell you, of course, that I unfortunately am no longer effective in my own lab. When I walk in there, they kick me out. They don't tell me where their cells are. So this could not have been done without the good postdocs and graduate students and people that are on my team. And as you also know, that in science these days, it's very hard to be really making any difference alone. So we work very closely with a number of collaborators across BU, Harvard, MIT, locally, and obviously some other places in order to get our work done. And then the last thing I want to just say is that it's also been really great to be part of this Biological Design Center, where I think we have a number of really exciting projects that are going on across a number of really great scientists here at BU. So it's a pleasure to be part of this community. Thank you.
So we have some time for some questions from the audience. Anybody? I'll start with one. The earlier slide in which you've got the cells to align and then be stronger and stronger depending on the stiffness of the pillars. What is, oh, hi everybody. What are the pillars made out of, and what kind of pillars are in the body that makes them do that?
Our pillars are made out of silicone rubber, PDMS. So that's why they're elastic. In our bodies, obviously the heart doesn't make a single fiber muscle, but is making a chamber. So in the body, what really provides that resistance is blood pressure. The valves and the blood pressure, right? As it's contracting, you get this return of blood that then expands the tissue. And that causes, I think, a great deal of alignment during that process. In skeletal muscles, obviously, it's the bone growth and the insertion points that does that. No need to be polite because, I mean, this is an internal meeting, right? So you can ask me all that.
So Chris, your method of generating microvascular is great, but can you generate organized beds of mesovascular that lead to microvascular and back out through mesovenules and so that it's useful as a little thing that can be
I
plugged
love the word
into
meso because you don't have to actually be pinned down on a particular scale. So what we find is that it's really hard to specify architectures that are at the 10 micrometer and lower scales. But fortunately, cells can reorganize things at that scale fairly easily. So at the next scales, right, 50, 100 micrometer, that is at the lower limit of what you can do with standard 3D printing methods. It's easy to do 0.5 millimeter and up. So there's this sort of one hierarchy layer that's hard. The upper ones are easy. You can get from aorta down to millimeters very easily. And the capillary bed structures are very easy. And it's just this one layer that's difficult. So Alice White, with the nanoscribe, I think has tools that can get us at that range. It's almost too high resolution, but I think there are ways to make that work. And then we're working with Jennifer Lewis, who's a leader in 3D printing over at Harvard, who also is trying to work at this length scale. So I think that some combination is going to make that work. Yeah.
Thank you. Really nice talk. I really like the idea that maybe mechanical force links the organization from the molecular structures to cell and tissue. And perhaps myosin plays a big role in this. Do you think that myosin may have a more general role in, like an evolutionary kind of role in the emergence of biological complexity?
Yes. But I don't have to prove it, right? That's your job. I don't know. I mean, I do think that if you look across kingdoms, there are many forms of myosins or other kinds of motor proteins that are really critical in all multicellular systems, from sponge all the way up. So absolutely. Other people would argue that the key evolutionary step was not myosin, it's a motor that's acting on filaments. It's really actin. And actin is everywhere, right, from bacteria up. So that could be the key factor. Once you have a filament, how you generate forces on that, there could be many ways to do it.
Chris.
Yeah.
So in that vascular patch you implanted in the mouse or rat, was that structured initially of the mouse's own cells?
Oh, that's really interesting. So we did these studies using human cells in immunocompromised mice and rats. We wanted to do that because we were thinking about sort of a development path where we would need to have human cells demonstrated all the way up. In the end, it turns out that I think we're going to need both. We need immunocompetent animals with syngeneic cells, right? Cells of the same source, because that's part of the sort of FDA requirements, and then we need to do human. What's really interesting is that the human cells ultimately disappear. So what we found is that in our implants, within about a month, you get this great vascularization from the host. You get chimeric vessels that have both human and host endothelial cells, and gradually the endothelial cells from the host, take over. Human cells are gone by the end of a month. So based on that, we're now thinking that we could potentially have a universal donor, right? Rather than having a matched donor for every person, right? Because then you'd have to get your own cells. Usually someone who needs this is going to have sick endothelial cells in the first place. So ideally, you don't want to use an old sick person's endothelial cells to help them recover from ischemia. So we could potentially use another donor, knowing that those cells are going to go away. then we just have to immunocompromise for a short period until those are gone. So.
Questions? Okay. Well, let's give Chris another hand. We have two gifts for Chris. One is this framed
Of myself, wow.
so he can, and we'll hang it on the wall, and then when the contractors come by, it'll like it fall and glass will break everywhere, like the other ones. And then we also have, in addition to the distinguished chair that he holds, he can hold a physical chair right here, engraved with his name on it.
Wow.
And this is the way we figured out how to give out lots of chairs to great faculty members that won't cost a lot of money. But congratulations and thank you very much, Chris.
Thank you.
Really
Thank you.
impressive.